Hello, dear students. As promised, I am recording the lecture on the histology of the nervous system. Let us begin with a short introduction. So we know that there are four basic human tissues. We've already discussed the epithelium, the connective tissue, and the muscle tissue. And now we are going to move on to the tissue associated with the nervous system. So as a general beginning, we know that there's a sensory input that is under the function of the nerve, and there is a motor output once that sensory information becomes integrated. So let us look a little bit more in depth as to what that means. So the nervous system is basically divided into categories. There are several different categories that we use. The first one is the anatomical division. The other one would be the functional division. So if we're looking at the anatomical division of the nervous system, it's quite simple. It's divided into the central nervous system, which is the brain and the spinal cord, and it's divided into the peripheral nervous system, which is basically everything else. And if we're looking at the division or the functional division of the nervous system, it looks like this. We have a sensory and a motor division. Sensory is the part that receives the information, and the motor division, which is the part that reacts. We're also called the sensory division the afferent division. Afferent is always integrating. The motor division is also known as the efferent division. Efferent meaning exiting or reacting. So we have a sensory division where we integrate and bring in that information, and the motor division, which is the part that will functionally react to whatever is being received. Now, the motor and the sensory division are also subdivided depending on whether it is under our control or under our perception, or whether it is not. So if we're looking at the motor system, we have two categories. One is the somatic category. That's the category that we are in control of, our motor functions that we are in control of, as well as the autonomic division, which are the motor controls that we are not conscious of, or we don't have any, I would say, control over. The sensory is also divided into both a somatic division, which is that sensory that we are perceiving, okay, we are understanding, we are acknowledging. That would be things like touch. And then there's a sensory division like the visceral division, which is also a type of sensory, but for functions that we are not even aware that we are having. So here are some examples. If we're looking at the somatic portion of the motor system, we are talking about things like the skeletal muscle, which is the best example. And if we're looking at the autonomic or the involuntary motor, I would say, functions that we are not in control of, we're talking about things like secretions from the glands or the heartbeat itself. And if we're looking at the sensory part of the nervous system, if it's under conscious perception, we basically talk about our skin. We talk about when we feel something, when we touch something, that we are aware of it, that we are perceiving it. But there's the visceral category as well, which is sensory things that we are not even aware of, things like the cardiovascular structures, like the blood vessels. We will receive a type of sensory input telling us to dilate the vessels. That is a motor function, yet it's a function that we aren't even perceiving that it is occurring, which is of course occurring constantly. One more thing you need to know about the motor system is that the motor system is divided into both an upper motor system and a lower motor system. And basically what it means is that we're talking about the neurons that are involved in messages that go from the brain to the spinal cord. And then we talk about lower motor neurons, which are the neurons that are located and receive messages from the spinal cord to the muscles. These are a division of the somatic or the the perceived and the in control motor neurons. Finally, the motor neurons are divided into something called the sympathetic, parasympathetic, and enteric divisions. This is the autonomic portion of the motor nerve. Okay, and so the sympathetic, these are the motor nerves, of course, that we are not in control of. And we divide it into something called the sympathetic and the parasympathetic, which are actually a balancing force one to the other. We'll talk about that in a minute. And there's something else that we're not even, we don't discuss very often, which is the enteric division of the autonomic motor nerve division. So anything that is related to stress, to crisis, to fight and flight falls under the category of the sympathetic autonomic motor nervous system. And anything that is related to either rest and breed or feed and digest would be considered the parasympathetic autonomic division of the motor nerves. And they work in balance together. When one is high, the other one is low. So if we're looking at anything stress related, okay, we're talking about the sympathetic system. And if you notice on this list, this diagram of the different functions involved in each, you'll notice that when we're talking about the stress related activities, anything that is required for us to get into quick action to fight and flight is involved in the sympathetic system. So anything that is non-crucial for that fight and flight gets shut down or slowed down, not exactly shut down. So anything involved in the GI or the GI or the urinary system, those are the two systems that normally become decreased during a stress-related activity or a sympathetic system-controlled activity. Things that are not necessary at that time for fight and flight become decreased. That is in opposition to the parasympathetic system. This is the system that takes over when we are resting, when we are in digestion, which is a type of rest, when we are feeding, and when we are breathing. And last on the list is the enteric nervous system. And this is also part of the motor system. It's a less talked about system, but it's very interesting because they also call it the second brain, because the nervous system that runs the GI is requires its own system due to its very complex and, of course, its necessity or for daily function. That would be the enteric nervous system. That would be things like digestion and sugar, et cetera, et cetera. So that would be the enteric nervous system. And it works completely independently of the rest of our CNS. Okay, so that was an introduction. It should be a review of your neuroanatomy and neurophysiology classes. But now we're going to talk about the central subject of today, which is the histology of the nervous system. Now, as complex as the nervous system is, and it's quite complex, things are being learned daily. We probably don't know even a small portion of what the nervous system can actually do. But if you break it down into histology, it's actually quite simple because it's made up of two primary cell types. It's made up of the nerve cell, which is also known as the neuron. And it's made up of the neuron's supporting cells, or its maids, known as the glial cells, also called the neuralgia. Anything that starts with neuro, of course, refers to the nervous system. So let us first take a look at those neurons. The neurons are quite extensive in the human body. There are probably over 100 billion different types of neurons. These are the structural units of the entire nervous system. They are excitable electrically, and their job is to propagate and generate a action potential. In other words, propagate and create a force that will allow for action. We have different types of neurons in the body, which we'll get into a little bit later, but they can have connections that run to the thousands, and they can be quite complex with huge branches of dendrites, and they could be quite simple with just a single axon coming off of the cell body. And we will look at that in just a moment. So let's look at the basic components of the neuron. It's made up of three basic components. The cell body, which I would say is the brain of the cell. That's where most of the important functions occur. Okay, that is where you would have your nucleus and the nucleoli and everything that is involved in the cytoplasm. And off of the cell body, you're going to get either an axon or a dendrite. Now, one of the important things to note about a neuron is it's amitotic, which means it does not have the ability to enter into the mitotic cycle, which means that they don't proliferate or repair. So once a neuron is damaged, it's usually permanent, but the neurons are also quite durable and they can last from birth to death. So in other words, a single neuron can exist for up to or over 100 years. Another term for the cell body you should recognize is soma or perikaryon, and the reason for that is that you might see it mentioned in different textbooks under different names. So here's your neuron. The neuron consists of the central brain of the system, or the cell body. Here within the cell body, you see a centralized nucleus. Inside the nucleus is the nucleoli. Surrounding the nucleus is your cytoplasm, and within that cytoplasm, of course, are all the components of every cytoplasm of all the different cells. Now, off of the cell body in one direction, you've got the dendrites, and in the other direction, you have the axon. Most neurons only have a single axon, but neurons can have anywhere from a simplistic dendrite tree to a very extensive type of dendrite tree. The axon can be short and it can be as long as the length of your body. In this particular case, you see that the axon is covered with something known as the myelin sheath, which we will talk about a little bit later. At the end of the axon is an axon terminal. The terminal is like a train station where you get off. It's where the last stop of the train is the axon terminal. And this is where the neuron will synapse with either another neuron or with a muscle or with even something like an interneuron, which is a neuron to neuron kind of communication, which we'll talk about later. So we're looking at that neuron, the neuron cell body, or the perikaryon, or the soma, how you want to call it. It's basically the same as most cells of the human body. It has the nucleus with the nucleolus. It has a cytoplasm with the different components like the Golgi apparatus, the mitochondria, the endoplasmic reticulum, et cetera, et cetera. It also has something unique, at least in the motor neurons, which we'll talk about soon, called the Nissl bodies. And these are basophilic granulations within the cytoplasm of the motor neuron. It also has something called intermediate filaments. Okay, like every cell of the human body, it contains its own specific intermediate filament, which gives the cell not only structural integrity, but it also gives us a way to recognize the type of cell that we are looking at under the microscope. So if we're talking about epithelium, for example, the intermediate filaments would include the keratins. And if we're talking about a muscle cell, the intermediate filaments would be included as the proteins within the intermediate filaments would be desmine and actin, for example. And if we're looking at the nerve, we also have its own intermediate filaments, which enable us to use immunohistochemistry and be able to highlight and recognize that we are looking at a nervous system cell under the microscope. Okay, so let's take a look at that axon. Like I said, the axon can be extremely long or it can be quite short. But what we do know and what's important about the axon is that it takes the information after it's been brought into the cell body by the dendrite and passes it along to its terminal, which will then synapse. These are your axons. The axons, if they're large enough and visible enough, are given also a name of nerve fiber. If you ever see that term used in your readings. Here's another example of that axon. It's coming off the cell body or the soma. It's extensive and it goes down to its axon terminal, and it will then synapse with another cell, which we will talk about in a moment. And here's your dendrite. The dendrite also, it can be quite extensive and beautiful, as you see here on the right. Lots of dendrites coming off the cell body, I would say entering into the cell body. And the reason for this extensive dendritic tree would be to be able to increase the surface area and be able to receive as much of the impulse as they possibly can. The more dendrites, the higher the chances of the strength of the impulse. Okay, because it is the dendrite's job to bring the electrical stimulation into the cell body so it can be integrated and passed on into the axon. Here are two cases or examples, diagrams of the cell body with its long axon coming off the cell body and its synapsing with a nearby motor neuron. As you see here, it's a motor neuron. And here on the right-hand side, you could see that the synapse is synapsing with a muscle. And the entire process relates to the secretion of neurotransmitters, which then go on to create another impulse that's received by the receptors on the other side of the cell, which will then release other chemicals and propagate the action potential, which you will learn in more in depth in physiology. Here's two examples of neurons under the microscope. Neurons are not easily visible under the microscope. They're way too small. Sometimes you can visualize it with an H&E if you're talking about a large motor neuron within the brain. But if you're talking about a peripheral neuron in the peripheral system, you probably won't be able to recognize it under the HE. It would require either electron microscopes to be able to visualize them. And in the central nervous system, you could pick them up with certain H&E stains or histochemical stains. But here we have two examples. This would be your cell body of the neuron. You can see these purple nissl granules or nissl bodies within the cytoplasm of the neuron. This is the central, in the center is known as the nucleus. The two dark dots in the center of the nucleus are the nucleoli. Here's another example on the right-hand side. You can very well visualize that it is a neuron. It has a central nucleus, a nucleoli, dendrites, and axons coming off the cell body. These little dots, okay, that are surrounding the neuron in both these pictures are the supporting cells or the glial cells, which we will talk about a little bit later. Next, we're going to move on to discuss the Schwann cell. The Schwann cell is very important because the Schwann cell is found in all of the different neurons. It is a type of glial cell. It is not a type of neuron, therefore it does not have the capability of propagating an action potential or passing on electrostimulus. But what the Schwann cell does is it's responsible for laying down that very important myelin sheath. The Schwann cell, as you see here in this photograph, you see the nucleus of the Schwann cell, and then you see that its plasma membrane just keeps wrapping around and around and around the axon, creating this type of almost like a toilet paper roll around the axon. And the Schwann cell is actually laying down what is known as the myelin sheath. The myelin sheath has both functions of protection as well as increasing the speed of the conduction. Okay, as the conduction passes over the myelin sheath, it increases in speed and moves on to the rest of the axon. Okay, so that's your myelin sheath and Schwann cells. Again, Schwann cells are a type of supporting cells or glial cells. And if we're asking ourselves, why are some nerves myelinated while others are unmyelinated? Why do some require this large rapid propagation while others do not? Well, we're not really sure. The factors that are the factors related to myelinization are a little bit unclear to us. But we do know that myelination is an important fact for propagating the electrical impulse. The next part of the axon that is required to know is the nodes of Ranvier. The nodes of Ranvier are here. They lie basically they lie where the myelin sheath ends and a new one begins. It's actually an area where the axon is exposed. In other words, it's not covered with this myelin sheath. And what we do know about these nodes of Ranvier is that they can have axons or collateral axons that actually exit from these nodes of Ranvier there are certain diseases associated with, I would say, closure of these nodes of Ranvier, which we'll discuss in pathology. This is an example of the nerve under the microscope. This is all you basically can visualize. This is a peripheral nerve, and all you can visualize under the microscope is, first of all, that it's a nerve structure. You can recognize it as a nerve structure. First of all, it's round, unless it's cut longitudinally. This is a transverse cut right down the middle, and here you can see that it's a nerve structure by its morphological structure, as well as the wavy, the wavy nerve fibers within this fascicle of nervous tissue. These little purple dots that you see here, are the nuclei. They're actually the nuclei of the Schwann cells. You can see this little space, this white space, which is the myelin sheath. The myelin sheath is composed of a lot of lipidous or fatty tissue. So during the preparation of the slide, a lot of this tissue kind of just dissipates out of the slide, and it's basically you're left with this empty little space. But this is what this area here would be the myelinated nerve, which is the axon covered with the myelin sheath. And here on the right-hand side, you would probably be able to recognize it as being unmyelinated. You do not see the space where the myelin sheath used to be. Here's another two examples. On the left-hand side, you see the unmyelinated nerve. You do not see the myelin. And on the right-hand side, you can see very clearly that each axon is surrounded by this white, right now it's white, but the myelin sheath that's surrounding the axon. And usually the little dot in the center, which you're going to recognize, it's actually the nucleus of the Schwann cell that gets picked up. But these are both H&E examples of nerve. The best that you can see here under the H&E would be that it is a nerve structure. In other words, it's a nerve, it's a fascicle of nerves. And you could probably pick up either the Schwann, the nucleus of the Schwann cells, and where the myelin sheath used to wrap around each axon. This is a closer view. It's not an H&E, it's a special stain, but what you can pick up here is that each of these represents an axon, and on the space that you see here is actually the node of Ranvier between the different components of the axon, where the myelin sheath begins and where another one ends. So this would be a good example of the node of Ranvier. Let's talk about the different neurons. Neurons are divided, or I would say categorized, either based on their function, which we discussed already, which would be afferent or efferent. Either it's a neuron that is responsible for the input or the export. And it's also based on its structure, If it's morphologically a unipolar, bipolar, or a multipolar type of neuron. And usually the categorization of the different neurons depends on the processes that extend from the cell body. So let's take a look at that. This is a beautiful example of a unipolar neuron. It's actually a pseudo-unipolar neuron. Most unipolar neurons are very, very rare in the human body. They're much more common in vertebrates or invertebrates. But here you can see that this would be your cell body, and there's only one process that comes off the cell body, and that is your axon. At the end of the axon, you do have these processions that come off the axon, but they do not contain dendrites or formal dendrites that come off the cell body. Here's a diagram of the same thing. You have your cell body with a single axon that comes off the cell body. This is a pseudo-unipolar, pseudo-unipolar. In other words, it looks like a unipolar. It's not an actual unipolar because of the way it splits down after it exits the cell body. But this is a good example of a pseudo-unipolar neuron. Here, it's either dendrites that it's attached to another type of neuron, but by definition, these pseudo-unipolar neurons contain only a cell body with a single axon. Usually this type of neuron is a sensory type of neuron as opposed to a motor neuron. And when the axon splits, you're going to get one branch that heads off into the CNS, while another branch will head over to the PNS. This is an example of a bipolar neuron, okay? A bipolar neuron meaning it has a single axon with a single dendrite. So this is an example of a bipolar neuron. It's a much rarer type of neuron in the human body, found in very exclusive areas like the nasal cavity. But just to recognize the bipolar neuron, remember it has, as opposed to the unipolar neuron, which has one process coming off of the cell body, here you see two processes coming off the cell body, 1 and 2. The most common type of neuron, though, is by far the multipolar neuron. This is a motor neuron. And the motor neuron is the one that we see in most cases in textbooks. It has a cell body with a long axon, in most cases myelinized, and dendrites on the other side. In fact, multiple dendrites. So because of these multiple extensions from the cell body, it's called multipolar neurons. Here's a diagram to show you the different types of neurons. Again, the unipolar neuron is mostly a sensory type of neuron, whereas the multipolar neuron is a primarily motor neuron. Here's an example of a unipolar neuron. Unipolar neuron are usually not located in the human. It's mostly invertebrates. When we see them, it's mostly pseudo-unipolar neurons, and those would be sensory neurons, things like the skin, the joints, etc. And then there's the bipolar neurons found in very isolated areas, things like the olfactory epithelium and the retina of the eye. These are beautiful pictures under the microscope with special stains, mostly crystal violet, I believe, which are multipolar neurons. A very large cell body and extensive processes that come off the cell body. These little dots, again, that are surrounding the neurons are your glial cells. Finally, the synapse. Okay, when we have the axon terminal synapse with something, it's either synapsing with another neuron, as you see here in this photograph, the impulse is being passed on to another neuron. These are actually interneuron neurons. And here on the right hand, the little box, you can see that it's synapsing with a receptor on a muscle cell, where it's releasing its neurotransmitters and propagating the stimulus. Okay, the neuron has the ability of either to excite something else or even inhibit the other thing. It doesn't necessarily mean to create an action potential. It can actually shut down the action potential. What's important about the impulse, though, is that it's always unidirectional. It starts at the dendrite, which gets passed on into the cell body. Okay, that's why it's called the afferent neuron. It's entering into the cell body being the base, the brain. Okay, so afferent to efferent refers to the direction from or to the cell body. So if the dendrite's coming into the cell body, okay, it's an afferent neuron, enters into the cell body, passes on into the axon, It's leaving the cell body. It's an efferent neuron, efferent, EFF, efferent neuron entering into the axon. Axon will propagate it over to the synapse, and of course the synapse then is where communication continues, always unidirectional. Okay, so this is what I just said to you. The signal will arrive at the dendrite, it will pass it on into the cell body. That's your afferent signal. The signal will then exit the cell body, get passed on into the axon and into finally the synapses. So that would be the efferent signal. It's exiting the cell body. There are different types of synapses as far as what is synapsing with what, and therefore it was given specific names. They're easy to understand because the axodendritic is exactly as it sounds. It's from axon or dendrite to axon. or axon to dendrite more likely. Axosomatic would be from axon. Somatic is always cell body, so it's from the axon to the cell body. And axoaxonal is axon to axon. So here are 3 examples of each. Axon dendritic, meaning the axon, as you see here, is communicating with another dendrite. On this one, axosomatic, you see that the axon is communicating directly with the cell body. And there's finally a synapse, the axon-axon, where you see the axon communicating with an axon. But of course, we all know the most common synapse would be from dendrite to cell body to axon, and then etc. Okay, let us take a look now at the glial cells. That was the first category of cells within the nervous system. That was the neuron. Now we're going to talk about the supporting cells, which is the glial cells or neuroglial cells. Neuroglial cells in the PNS are a bit different than the CNS. The PNS, we have 2 cells, the Schwann cells, which we discussed already, as well as the satellite cell, another type of supporting cell. In the CNS, we have different cells depending on their function. One is the oligodendrocyte, the other is the astrocyte, also known together as macroglial cells. And then we've got the ependymal cell as well as the microglia cells. Okay, so what is the function of these glial cells? First of all, it's important to know that there are many more glial cells than there are neuron cells. The glial cells, which are supporting cells, run in the hundreds of billions within the central nervous system and the peripheral nervous system. Remember, these are not neuronal cells, therefore they do not have the ability of propagating an electrical stimulus. They're just there to support. What does that mean, to support? First of all, they maintain homeostasis in the whole system. They form the myelin sheath, for example, the Schwann cell. In the central nervous system, the oligodendrocyte. It provides support and protection for the neuron cells. It supplies the nutrients and the oxygen for the neuron cells. They insulate one neuron from the other, and a type of glial cell has a specific function of destroying pathogens and removing dead neurons, the microglial cells. So you can see that without the glial cells, the neurons would never survive. It's up to the glial cells to make sure that the neuron is protected and functioning the way it should. Okay, so we talked already the Schwann cells. Is just to remember, it is a glial cell. It's found in both sensory and motor ganglion. Here are the Schwann cell nucleus. Now, even though all cells have Schwann cells, not all will create or produce the myelin sheath. I think this is an example of an unmyelinated nerve, a bundle of nerves actually, and the nuclei of the Schwann cell are clearly designated on this example. So if you're going to see the nucleus in general, it's the nucleus of the Schwann cell, at least on an H&A. Okay, the satellite cell, we're not going to go into very deeply, just to note that they are found in around the ganglion. These cells around here are the satellite cells that are found surrounding the ganglia in the PNS. Remember, satellite cells belong to the peripheral nervous system. Here's a nice example of the nerve. This is a nerve cut longitudinally as opposed to transversely. So it's almost cut along, like a carrot, along the length of the carrot, as opposed to slicing it into little pieces horizontally. And what you notice, I think, is that this nerve has a very wavy type of appearance. That is very, very characteristic of a bundle of nerves. It's not a single nerve. This is a bundle of nerves. Each one of these nuclei represents a different neuron, okay? And each one of these nucleus most probably is a Schwann cell surrounding the axon of each neuron. So this is what you see here. You see this entire structure is a nerve made up of multiple nerve cells or neurons, including their Schwann cells and their myelin sheaths in some cases you see here. And here, the white area would be your myelin surrounding each axon. Okay, the nerve sheath. Before we head into the CNS, let's complete the peripheral nerve system by talking about the nerve sheath. Now, just like we did in the muscle, where each muscle fascicle was surrounded by a perimysium, and each individual muscle cell was surrounded by a endomysium, and the entire structure with an epimysium, same thing with the nerve tissue. For looking at the entire nerve tissue, okay, we're talking about several fascicles of bundles of neurons. It's covered by the epineuron. And if we're looking at each fascicle of the nerve, we're talking about a perineuron. If you go way, way inside, which you can't see at this light, at this power, each neuron is surrounded by an endoneuron. Okay, so this is the basic structure of the nerve tissue. Here are more examples of peripheral nerves that are under the H&E. I told you that under the H&E, you will not be able to recognize all the different compartments of the nerve tissue. All you'll be able to really recognize under the H&E for peripheral nerves is these fascicles and bundles of nerve tissue. As you see here, all this is transverse cut as opposed to that longitudinal cut. Here at the bottom, you can see a little higher power where you're looking at within the fascicle. And as you look here inside, you're going to see the Schwann nuclei surrounded by the myelin sheath, okay? And around each one of these little circles, you'll find the endoneurium. If you're looking at the right-hand side, again, this area surrounding each one of these circles is the endoneurium. And within each of these endoneurium is an axon, which has a Schwann cell and a myelin sheath surrounding, at least in this example, surrounding each axon. Here's a nice picture again of the peripheral nerve. On the right-hand side, again, cut sagittally, not longitudinally. You see a large fascicle of nerves, okay, of axons, and inside are each individual axon surrounded by endoneurium. But here too, you can see this wavy type of, I would say, structure, which is representative of a nerve, this wavy type of configuration. And if you're looking at the left-hand side, which is a comparative picture of nerve from the muscle tissue, here you have skeletal muscle fibers. And on the bottom left here, you have a nerve. The nerve, again, can be recognized by its wavy appearance, its centralized nuclei. In most cases, you'll find some myelin sheath surrounding the neurons. And on the right-hand side, the skeletal muscle, of course, if you remember from our second class, has a nuclei that is at the edge of each cell. Here's a diagram again of the three sheaths that surround the individual parts of the nerve. You have the epineuron surrounding the entire nerve. You've got the perineuron, which surrounds each fascicle, and inside you'll find the endoneurium surrounding each individual axon. This is also the same type of picture, if you notice, very similar to what we saw when we studied the muscle, the same type of configuration. Okay, here again is the epineuron surrounding each fascicle of the nerve tissue. Okay. Again, here at the covering the entire nerve tissue or nerve trunk is the epineuron. Surrounding each individual fascicle, you see perineuron, and surrounding each individual axon, you'll find the endoneurium. Again, the same type of thing, just to make you understand clearly exactly what is meant by endoneurium, perineuron, and epineuron. The last thing that we're going to discuss, it's a interesting thing. It's called the Renault body. And the Renault body does not have a known function, but it's definitely there in the nerve. We have yet to figure out what its job is, but there are these hyaline bodies, which are like a dense eosinophilic type of body that protrudes out of the perineuron and extends into the endoneurium. These are Renault bodies. function unknown. Now we're going to finally move on to the central nervous system, or at least the basics of the histology of the central nervous system. We're going to talk primarily of the cerebrum, the cerebellum, as well as the spinal cord. Again, the cerebrum, the cerebellum, as well as the spinal cord, three components of the central nervous system. Now, the whole subject of neurons in the brain is quite complex, whereas we know that the basic types of neurons in the peripheral nervous system, when it comes to the brain, there is still much that we don't know of. We do know that there's, of course, motor neurons, which are multipolar neurons, as well as the pseudo-unipolar neurons, which are sensory. But as far as a complete understanding of the brain and its neurons is still very much a mystery to all of us. Let us discuss what we do know. So these are, again, these large motor neurons. You can recognize it with the nissal bodies. Here we have the nissal bodies, both cases. It's in the cytoplasm. They're basophilic and they're very granular. So it's easily recognized as the nissal bodies within the cytoplasm. Now, what are these nissal bodies? They're basically composed of a lot of ribosomes on top of the rough endoplasmic reticulum. And of course, the function is protein synthesis to create different proteins, which are obviously very much needed when we have such an active neuron in the motor nervous system. Okay, this is a special stain, the crystal violet stain, which really highlights those basophilic nissal bodies. Here's another shot of those nissal bodies. You can very clearly see the nissal bodies within the cytoplasm of the neuron, as well as here, surrounding each neuron, you can see the glial cells in both examples, which are, of course, the supporting cells of the neuron. One of the interesting cells within the central nervous system are the interneurons. And these interneurons are there to kind of like communicate between different neurons. Okay, so for example, here on the right, you see a sensory neuron, which needs to integrate with the motor neuron, and it uses this interneuron, this intermediate or mediator, to help communicate between the sensory and the motor neurons. Okay, so they connect and they transfer the signal from the motor or between the motor and the sensory neurons. They also enable to communicate interneuron to interneuron. And what's important about the interneurons is they are motor neurons or multipolar neurons or somewhat motor neurons, similar to the multipolar neurons that we've studied up until now. Okay, let's move on to the glial cells within the CNS. The astrocytes, which provide almost all the functions for the neuron. They're the ones responsible, the most numerous within the CNS as far as the glial cells, and they give all of the support and nutrients and oxygen, and they provide all the needs for the neuron. Those are the astrocytes, those are the main guards. Then you have the oligodendrocytes, which are responsible for basically laying down the myelin sheath around the axon, similar to the Schwann cell of the peripheral nervous system. The microglial cells are the cells responsible for increasing the immunity for these neurons. Okay, they're the ones that roam around and search for any pathogen or toxin that doesn't belong within the nervous system or within the nerve, as well as I guess relieving the nervous tissue of anything that has died out and requires to be excreted from the area. Now, those are a type of macrophages in actuality. That is what a macrophage does. The macrophage roams around tissues, finds things that don't belong, either something dead or something that is toxic, and will swallow it up and exit it from the body. Then you've got the ependymal cells. These are special cells. And they're the ones responsible for creating the spinal fluid that are cushioning the neurons. Okay, so those would be your ependymal. So these are 4 examples of glial cells within the central nerve system. The astrocytes and oligodendrocytes are known as the macroglial cells because of their size and distribution. And you've got the microglial cells as well as the ependymal cells. So let's begin with the oligodendrocytes. These have a shape more reminiscent of something round. Again, they're the ones that will lay down the myelin sheath. They will wrap themselves around the axon and create this myelin sheath in the axons of the CNS, the oligodendrocyte. The way you can recognize the oligodendrocyte in the CNS is the same way you can see the Schwann cells in the PNS, and that is because it is surrounded by this white halo, which is representative of the myelin sheath. So this would be your oligodendrocyte, and just like the peripheral nervous system, the myelin sheath is composed of a very fatty lipid-like tissue, which during preparation of the slide gets removed or dissipates or falls out of the slide, and you're left with this empty space around it. So this would be your oligodendrocyte, a large round nucleus surrounded by a halo of the myelin sheath. On the left-hand side, electron microscope, you can see this beautiful round oligodendrocyte wrapping itself around an axon here and here and here, actually. Beautiful, beautiful photograph. The next on the list, which is the most common, is the astrocyte. The astrocyte is responsible for all of the functions and all of the necessities required for survival of the neuron. It has a special shape, which is why it's known as astro meaning star-shaped. It has a star-shaped type of appearance due to its very extensive dendritic processes that come off the cell body, giving it a star-like appearance. Here on the left-hand side, you can see it as well. These very, very, I would say, these thin dendritic processes coming off the cell body. Here you can see that they reside very, very close to the blood vessels within the brain. Of course, the reason for that would be to maintain this blood-brain barrier, which will not allow anything foreign to reach the brain that would be under the guard of the astrocyte. Now, there are really two types of astrocytes. We have the fibrous astrocytes as well as the protoplasmic astrocytes. We'll get into the composition of the brain, but to give you a aforementioned, in order to talk about the astrocytes, the entire system is divided into a gray matter and a white matter. White matter is white because it has numerous myelin sheaths, and the other type would be the gray. So the fibrous astrocytes are found within the white matter. Those are the ones responsible for maintaining the blood-brain barrier. And the protoplasmic astrocytes are located in the gray matter. You can recognize them because they're a little bit wider. They're not as delicate as you saw in the previous picture, where the astrocyte had very fine extensions of dendrocytes coming off of the cell body. We'll show you an example. So this would be a case of, again, there's the blood vessel, it runs straight across this tissue, and the astrocytes, multiple astrocytes surrounding the blood vessels. The astrocytes are different than the neurons, and they're not as large, and at least the multipolar neurons are not as large and extensive as the multipolar, but you can see that under the microscope at this power, they have a star-like appearance. Okay, and these surround the blood vessels to maintain that blood-brain barrier. Now, in comparison, if you've got the fibrous astrocyte up here looking in the left-hand side, looking like a star, down here, it's also a star-like appearance of the protoplasmic astrocyte, but you can see that the processes are somewhat broader. It's not necessary to be able to recognize the difference between the two of them because it's very subjective. Just know that the two types exist, one in the white matter and one in the gray matter. The fibrous astrocyte in the white matter, the protoplasmic astrocyte located within the gray matter. Next on the list of glial cells is the microglia. Again, they act as macrophages to maintain I would say, the immunity of the neurons within the nervous tissue. They don't let anything get in that doesn't belong in, and they make sure that if you do enter, you'll be swallowed up and taken out of the room. Finally, the ependymal cell. Now, the ependymal cell, when you first look at it, looks like an epithelial cell. It looks like a low columnar epithelial cell, but don't be fooled. It is not an epithelial cell. It is a supporting cell or a glial cell. Its job is to create the central spinal fluid for cushioning these neurons in a nice, warm, comfortable environment. It's found in the spinal cord in the brain. And they have also cilia on their surface, which kind of keeps things moving along, circulates that CSF or the spinal fluid in the CNS and keeps it, I guess, flowing. Okay, so now let's look at the specific tissues, the spinal cord and the brain. The spinal cord and brain are both made up of gray and white matter. There's only one big difference. In the spinal cord, the gray matter is on the inside, the white matter surrounds it, and in the brain, it is the gray matter that exists on the outside and the white matter on the inside. So here's an example of a spinal cord with the gray matter on the center and the white matter surrounding it. Now, both the gray and the white matter, they both contain the oligodendrocytes for the ones that are responsible for laying down myelin and the astrocytes. In the white matter, though, okay, you will not find neuronal cell bodies. All you'll find are myelinated axons. That's the white matter. That's what gives it the white look, the myelinization, the myelin, okay? So it's myelinated axons within the white matter. Within the gray matter, you're going to find lots of neuronal cell bodies, but you're going to find that the axons are unmyelinated. That's why it's gray. It doesn't have that white myelin sheath surrounding the axons. Here's an example of the spinal cord again. Now, this gray area is also split by its zone. Some books refer to those different terminologies depending on the different books you use, but basically it's divided as a dorsal root. That would be the root facing more of the back. You've got the ventral root. That would be the root or the horn that is located more in the interior part. And then if you want to subdivide it also, you get the lateral root. That's the one on the side. So you have the dorsal root, the ventral root, and the lateral root or horns, depending on how you want to call it. Okay, so again, here's your spinal cord under the microscope. You see beautifully the gray area or the gray matter here in the center. It has an H type of shape. You would have the dorsal root or the dorsal horn over here, the ventral horn over here, the lateral horn would be over here. You would see in the center, the central canal. That is where the ependymal cells will be located, the ones that are responsible for laying down the spinous fluid. Here on the outside, you're going to have the white matter, recognized very easily by its white appearance due to its rich myelin on the axons. Again, no neuronal bodies, but a very nicely myelinated axons here in the white matter. Here's a closer view, the same principle. You got the dorsal horn, the ventral horn, the lateral horn, and a central canal filled with the spinal fluid. A closer view of the central canal with its glial cells surrounding it. The glial cells known as, of course, ependymal cells. And here's one more photograph of a diagram showing you the different horns within the gray zone or the gray matter and the myelinization here in the white matter. Okay, now the dorsal horn of look at the gray matter of the spinal cord. We have a dorsal horn, which are composed, that's the one that we saw up on top. That would be, they contain more of the sensory neurons. The ventral horns, and you can see it beautifully, these large multipolar neurons here within the ventral horn. So if I'm going back 2 slides, I'd like to see, you can see it here, that the dorsal horn, okay, is located here, and down here in the ventral horn, you can see very clearly this rich number of multipolar motor neurons. Okay, so again, this would be your gray matter, your dorsal horn, which contains primarily sensory neurons, and the ventral horn containing primarily your multipolar motor neurons. Okay, here's another view, beautiful view of the ventral horn of the spinal cord with its multipolar neurons. You can see the nistal bodies within the cytoplasm. You can see the glial cells here in the background. You can see some oligodendrocytes surrounded by the myelin sheath. In fact, they're primarily oligodendrocytes with some wandering microglial cells. And here on the right-hand side, you can see the difference between the two is the beginning of the white matter. Okay, one more time, you've got the multipolar motor neurons with its glial supporting cells, the white matter on the right, which contains only myelinated axons without any neuronal bodies. And again, you can see very nicely the oligodendrocytes. Okay, multipolar neurons and the nistal bodies within, a nice long axon with some dendrites, and on the right-hand side, your white matter. Next, we're going to move on to the cerebellum. The cerebellum has a very interesting conglomeration to it, and the point, of course, is to be able to increase its surface area. But the cerebellum, what's important is it also has a white and gray zone similar to the spinal cord, the difference being that the gray zone is now on the outside and the white zone is now on the inside. So where the arrows is pointing would be your white zone or your white horn or your white matter. And on the outside of the white matter is your gray matter. Now the cerebellum is composed of three separate layers, at least in the gray matter. It consists of the molecular layer, the middle layer, and the inner layer known as the granular layer. And the middle layer is also known as the Purkinje layer because of its Purkinje cells. Okay, so again, you'd have the molecular layer out here, the middle layer with Purkinje cells, which is difficult to differentiate, and the inner layer, which is composed of these very dark granular, it looks granular because these packed in small neurons. Okay, so you'd have, again, the molecular. Up against the inner granular layer, you'll find the middle layer, or the Purkinje layer, and finally this granular, highly packed, dense neuronal layer, which is the inner granular layer. Here's a closer up view of the molecular layer. Here you can see these Purkinje cells. They're very, very large neurons. And down here is the granular layer, which are simply small packed in neurons. Here's another example of the Purkinje layer with the Purkinje cells. They have this very unique appearance, almost like an antler on a reindeer, okay, which reaches into the white matter. Okay, now we're going to move into the cerebrum. The cerebrum also is divided into layers, but unlike the cerebellum, which is pretty clearly designated, you can see the layers almost clearly. This has 6 layers, not 3 layers, but it's very difficult to be able to put a line and designate where each layer begins and ends. Okay, so it's not necessary to memorize all the different 6 layers, just to know that, again, the gray zone or the gray matter lies on the outside, similar to the cerebellum. The white matter lies on the inside, and the 2 layers are composed of 6 layers of different cells or different layers, which contain different amounts of different types of neurons and things like that. Okay, but if you're looking, one thing you need to know about the cerebral cortex, that within the gray matter, you've got a cell that is classically, I would say, known for its position within the cerebral cortex, and those are the pyramidal neurons. And the pyramidal neurons, again, are large neurons, motor neurons with a pyramidal shape, which is why it was given this special name known as the pyramidal cells. And these are your pyramidal cells. You can see the nistal bodies within the cytoplasm. Again, they're multipolar neurons, and surrounding, you can see a few of these glial cells. Well, that's it for today. The nervous system is an exciting system with lots of new information coming to us all the time. So I want to thank you for your listening. I hope you enjoyed, and we will meet up again on our next issue of Histology.